Okay, thank you. My name is Weikang Ma. I'm the Beamline scientist that responsible for the fiber diffraction program. So, a large chunk of the fiber diffraction is muscle diffraction. So muscle it's kind of has this unique structure that is naturally a crystal. So if you shine a very bright X-ray, you can get the structure information at the molecular level from the muscle, and which has been widely used for decades. The first paper that using this technique is 1953. So it's a old technique, but with new capabilities from the synchrotrons that has come to new life in recent years. So it's much widely used recently in the community. So the reason it survived for 70 years, 80 years, because it has still some unique features that only possible use this technique. So it can provide molecular information at nanometer scale. It's not technically high resolution, but is very useful at this resolution. And importantly, you can collect data under physiological conditions. You can get from permeabilized muscle or even from live muscles, or you can. For insect, you can show the entire insect in the beamline, and you get the diffraction pattern while the bugs flying or something like that. And also, you can do time result experiment that you get the structural information under physiological relevant time scale down to sub-millisecond. So we like to see that X-ray diffraction is the only technique that allows simultaneous collection of global structure and the physiological information on this time scale. So, and most recently is in last decade also widely used for basic biophysical characterization of myopathies. So those are the old information. And also back to the old days, each investigator brought the entire boxes of the equipment to the beamline and spend days of beam time assemble their equipment. And then if you have 6 days beam time, you probably need to spend 3 or 4 days to testing stuff, synchronize, analyze stuff, and then the efficiency is much lower back then. Then since I took over a long time ago, 8 years ago, and we kind of replaced or we slowly equipped all our beamline with all the Aurora physiological apparatus for transducers and the motors. Then over the years, we accumulated 6 different Aurora 400 level force transducers and the 5 different motors and 3 dual model muscle levers for intact studies and 2 Linux, 2 Windows data acquisition systems. So basically, we have backup on everything. So in case something fails, and we have laser diffraction systems, and we have 2 3D printers for quick modification of the setups. So if you look at the picture here, all this red part or the transparent white part are all 3D printed. So if you need a specific change to accommodate your own experiment, we can do on-site modification quickly. And also we have this setup that put together in parallel, so you just slide things in and out. You have multiplex system that significantly increase the throughput. Those kind of system is especially useful for drug incubation. If you need to wait 10 or 15 minutes between shot, so the waiting time is gonna be very low efficient if you only got one system there. So, but use this, you can basically avoid that. So I don't know how many of you visit different beamlines over the APS down. I visit several beamlines. One thing that is quite challenging working on the beamline is the realistic. So there's not enough space to doing the stuff. And for muscle X-ray diffraction, it's not a simply data collection. It's more it's performing an experiment that need a large group. You need several people to perform the muscle X-ray data and data collection or perform the experiment. So space is always a limitation. So we have a fully equipped wet lab, but over the break we kind of repurposed a storage space that we installed the lab benches, and so you will have more spaces for dissection or for sample preparation. And also we acquired a Aurora myosite system that can be used by the users for to collect the mechanical or contractility data from the same sample by the same investigator. This kind of equipment is quite useful, especially for the kind of experiment that the sample quantity is limited. For example, we have users that doing biopsy studies. The sample is kind of they only got that much sample, and X-ray usually the biggest consumer. Whatever left over can be readily used on this myosite setup that you can collect the other biochemical or functional data from it. So those things is I still need to tidy it up, but it's readily available for users to use. Another thing is this isoflurane machine that we received a couple of years ago, but it took quite a while to get a safety protocol approved by APS. Now it's fully approved and ready to use. So we expect that this machine gonna greatly reduce the animal usage for, especially for intact skeletal muscle project, because we can keep the mice alive for the duration of time, so we can extract as many muscle as possible. So back to when I was doing this kind of experiment, we can only extract one muscle from one mice. And by the time you're done with the first experiment, the second one probably not useful. So this is the second thing. So we are running a user program right now with about 20 active users from domestically and internationally. So as some of you wonder how to get beam time, or how your data collection is done here at BioCAT. So we have three kind of access mode. The first one is the traditional mode that you got a large group, that you have a group of people with that with experience and some sort of experience on X-rays, and you come back, we get the setup ready for you, and we say good luck, and you can do everything you want to do, and just call us if you need any help. That's the traditional mode. Then there's a hybrid mode that so basically over the years the groups with limited resource or limited personnel, that they send one or two people that know their system the best, and then they collaborate with me and another student that at the Beamline, so we can collect data together. The third one is fully remote collaboration. So basically that's you sample, the users send the samples to us and then we do the experiment. So it's mostly used as a feasibility demonstration tool. So basically for first time users, they want to see whether their sample is good for X-ray diffraction. So we do some preliminary data for them. Or it's under very special circumstances. For example, some of our other collaborators doing muscle structure study, but they don't really look at things at fiber level. So they don't have people in the lab to dissecting fibers. So we also do a small portion of this kind of experiment. But we were limited by the personnel at the Beamline. But last couple of years, we won a couple R1s, so we are building up the academic lab that kind of independent with BioCAT. So we expect we'll have more capacity to take more remote collaborations and hybrid mode collaborations. So the things that affect fiber diffraction the most, probably the beam size. So this is the pre-upgrade beam size. You have much bigger horizontal beam size, that in order to better resolve the crowd structure on this along the monofilament, we want to put the muscle in vertical direction. So basically we take advantage of a smaller vertical beam. That In some cases, it's inconvenient, inconvenient to put things in vertical. But now the beam shape is actually smaller in horizontal orientations. We don't really need to put the muscle vertical anymore, and we can fully resolve the very crowded structures on the meridian. This is the picture from pre-upgrade. So basically we use this similar camera lens, same detector. So you see, this is the very crowded meridional M1 cluster, M2 clusters. So you see that it's more or less separated, but post upgrade, if you look at the separation is much better. So if we zoom into this M2 cluster, so pre upgrade, the bottom 50% intensity kind of lumped together. But you look at the post upgrade, we can at the same camera lens, same everything. Yes, you can almost fully resolve those very crowded reflections. Another one is in-air shot. This one is developed by the colleagues in Europe. They've been using for a while, but we haven't implemented it at BioCAT. But recently we kind of adopt this technology. We put the entire system on a translational vertical stage. So basically, you just align your muscle and then just take the muscle out of the solution, and then take the shot and put the muscle back. And we took so this is the comparison in solution and in air. So the difference is pretty obvious, as you can see. So just the basic estimate, we say that the diffracted intensity about 3 or 4 times stronger in air. So there are some optimization we need to do. So for example, we want to know how long we can keep the muscle in the air, or we can have a steamer stream and that keep the muscle moisturized, or how we better control the temperature once we take the muscle out of the solution. But overall, we think this is going to be the routine setup for our future muscle X-ray diffraction study, that we're going to collect the data of, in air to get much better diffraction qualities. And this is about the new stuff. We are still doing the permeabilized and the intact skeletal muscle study that I didn't talk about much, but we're still doing the things that we used to do. That's it. Thank you.